components of our system, shall we? So you've got the membrane protein M, which is this little guy here, shown upside down. Gross, but they do always show it upside down. I don't know why. Somebody made a decision. Good for them. That's going to be going through the membrane 3 times. These little squidgy bits that look like fingers at the end, those are the 3 transmembranes. And M is going to exist as a dimer. So there's always 2 copies, so there's always 6 transmembranes there in a bunch. N, nucleoprotein, is going to fold up. It's going to end up on the inside of the virion, touching the RNA, probably with the RNA wrapped around it. And the N is going to interact with M and the RNA. It's going to dimerize, so there's always 2 N's going together. Yeah. Those are going to dimerize via the carboxyl terminal domain, or CTD, is how it's going to be abbreviated in these slides. The E protein, it's not really in the virion. You'll get a couple of them squeaking in, but mostly E is there. Once the virion has budded, E comes in and cuts them off, and it looks like a few of them may get sort of stuck in there, or may accidentally hop on, interact with an M or something, and then just get lost in the shuffle. E protein is tiny. E protein can get lost in the shuffle. That's E. On its own, E forms a pentamer, a little 5-sided ring that could let ions into the virion. If it's there, it may help a little bit, but people have shown that it actually doesn't matter for the efficiency of entry. So, yeah, it's probably not important. Or rather, the ion channel activity of E does not matter for entry. The last one is the spike protein. Spike is shown here. There are little green dots on the outside of this one instead of little red dots on the outside, like in the model that you're thinking of in your head. And S is for attachment. S barely goes through the membrane. It has a little transmembrane at the end of each S that goes through, and then it'll sort of curve around, has a little acylation that'll hook back in. But almost all of S is floating above and outside the virion. So S is going to depend on M for incorporation. S interacts with M. S is a trimer. There's 3 of them. And S makes no contact whatsoever with the nucleoprotein or the RNA, as far as we can tell. And nobody has even been able to show any real contact between S and E. So S is depending on M to get it to the right place, and that seems to work most of the time. You can actually make virions without S. And that's the sad, cold truth. The virion is made, budding, scission, all that stuff happens just fine without S. S is incorporated if it's present, but S doesn't have to be there for the party to start. So, yeah. Sorry, S. Sorry you had to hear that from me. All right. So let's talk about some of these things with expression first. So, if we're going expression, what are the modifications, post-translational modifications that have to happen to these things? So for the SARS coronavirus M protein, we're not sure if it has O-linked glycosylation, but for most closely related beta coronavirus M proteins, they do have O-linked glycosylation. So you can pencil it in as that, but it is currently not actually known. And if you're watching this in the future and it is known, good for you. You got one up on this guy down here in the corner. This guy, yeah. Let's see. Yeah, so the glycosylation, if it exists, happens before the first transmembrane of M. Nucleoprotein gets heavily phosphorylated. It phosphorylates the heck out of that middle domain, which is the SR-rich domain, which, like a goofball, somebody has called the N3 domain here. Don't do that, people. It's the SR-rich domain, which they're differentiating for some reason that I don't understand. And here's some S's and some R's. And so it's the S's that are going to get phosphorylated, and that's going to basically cover up the extra charge that would be there on the R's. Yeah. There are probably other functions for this. Other functions have been proposed for this, like other reasons why the virus needs to do it. I'm not confident enough about any of those to put those into a lecture yet, but yeah, there's probably a reason. E protein is going to be palmitoylated, which means we're going to add a palmitic acid, a little fatty acid, to the tail on some cysteines right after it goes through the membrane. And that's exactly what happens to spike as well. Spike also gets palmitoylated on cysteines just after the transmembrane. And spike is heavily, heavily, heavily glycosylated, end-linked glycosylation at 22 distinct sites, each one with a somewhat different version of N-glycosylation. So they all start out, they're all attached to an asparagine, so it's N-linked, but there is a characteristic like length and fullness and number of modifications on every single one of those glycosylation sites. And you know what? It probably makes a difference somehow. Right now we know they're different. We don't know why or what it means that they are different, but they are different. So there you go. And no, you don't have to learn what each different one is for the test. Yeah. All right. Next part of expression. Let's talk transport. How do these things get where they need to go? Sometimes we know, sometimes we don't. All right, so the M protein is going to interact with Golgi and ER to Golgi intermediate compartment complex proteins via recognition motifs near the C terminus. Those are what the words say. In some coronaviruses, it's the same sort of motif, like a dibasic motif, like a KXHXX, something like that, that you would see used for the spike protein down here. For SARS-CoV-2, we actually don't know what the motif is. Isn't that embarrassing? We don't know on this well-studied coronavirus. But for other coronaviruses, it seems to be that. It's interacting with proteins, and it is transported actively to the Golgi and ER Golgi intermediate compartment. Nucleoprotein gets to come along for the ride if it interacts with an M, but nucleoprotein probably does not actually get trafficked to the right place. Nucleoprotein tends to hang out in the vicinity of a DMV pore, and we're not sure if it's actually transported there. Some of the phosphorylation may just be a way to get it transported to the sites where it's needed. That part is not known. E protein is going to interact with ER proteins, and it is going to help to retain S in the endoplasmic reticulum. So E and S and some endoplasmic reticulum retention proteins are all going to interact. That's not completely known, but M is also potentially involved in this process. So spike has to get 22 different sites glycan. It's like getting 22 different haircuts at one salon. It's a lot. It's really a lot. And so these 22 different glycans that are blown out and then cropped back are going to need some time. And so E and M are going to help slow S down so it gets glycosylated right. And so they're going to hover between the ER and the Golgi all by themselves through interaction with ER proteins. Again, we don't know exactly which ones for SARS-CoV-2, but we do for some other coronaviruses. That's why I'm able to say the general type of protein. The spike has a KXHXX motif. That's going to interact directly with cotomer type 1. That's what this COP looks like an I thing is. There's a cotomer type 2 as well. That's involved in a different type of transport in a different part of the cell. Cotomers form coats around a little vesicle, and the vesicle is going to have cargo proteins on the inside of it. It's just how stuff gets moved around inside of a cell. Cotomer has a bunch of different subunits. Cotomer beta is the one that most directly interacts up here, but you can see all the different ones. They each have their own Greek letter. And the cotomer beta and alpha are going to interact with these WD-40 motifs, which have, I believe, tryptophan and aspartic acid in them. Tryptophan is W, aspartic acid is D. And they're sort of repeats of those. And those are going to interact with the KXHXX motif. And that's how spike is going to get retained at the... it's actually going to get trafficked between the Golgi and the ER. Every time COP1 locks onto it, it gets dragged back to the ER. Every time COP1 lets go, it's going to move its way right back to the Golgi. And so in the Golgi, we're going to trim those glycans. In the ER, we're going to build those glycans. And so it's just back and forth until we are salon fabulous and ready to go out into the world. And then spike does. But basically only with its entourage of M and E and N and the genome. We call it a virion, yeah. And some lipids. All right, so That's the expression part. Now we're going to go through the other parts of this, and I'm going to do one part for each of the proteins because they take a minute and it's easier to watch small YouTube videos. Yeah, that's what the algorithm says. Okay, see you later.